Next is tertiary structure. Next slide. For example, in enzymes, which are globular proteins, how the entire chain, a very big chain, the polymer, folds itself several times and remains in the form of a ball-like or a globe-like. So there are various factors which contribute for the stability for the protein to remain in that particular form. So that is referred to as tertiary structure. What all factors which contribute for the protein to remain in that form? What all factors which stabilize for the protein to remain in that particular form? Important ones are the disulfide linkages, hydrogen bonding, hydrophobic interactions, linkage between oppositely charged R group, for example, aspartic acid COO minus, lysine NH3 plus. So an interaction between these two groups and weak van der Waals forces, all of these stabilize the protein to remain in that form. That is tertiary structure. Quaternary structure refers to such a protein which is made up of 2 or more polypeptide chains. For example, hemoglobin. It has four polypeptide chains: alpha 1, alpha 2, beta 1, beta 2. So in the diagram, you can observe the 4 polypeptide chain, how these 4 polypeptide chain are coordinately linked to one another, associated with one another to remain as one system or one protein. That is referred to as quaternary structure. So peptide bond is the one which links the amino acids in all the proteins and polypeptides and peptides also. So I told you in the beginning, these biomolecules generally formed by condensation, polymerization. There will be elimination of water when the monomeric units join together. So here, when 2 or more amino acids join, there will be elimination of water. The interaction occurs between carboxylic group of one amino acid and amino group of the other. So OH from COH is eliminated, hydrogen from NH2 get eliminated. So HOH combined to form water, and C double bond O NH is the one which links the 2 amino acid. So that is the bond between carbon and nitrogen as shown here. That is referred to as the peptide bond, the nature of which is amide bond. It is chemically an amide bond, C double bond O NH. So the important function, so the major function is they act as biocatalysts. You know what is a catalyst? A catalyst is a substance which alters the rate of a reaction without undergoing any change by itself. So that is a catalyst. Like inorganic catalysts, the biocatalysts are nothing but proteins. Here, lipase is given, which is involved in hydrolysis of the phosphodiester bond in lipids, hydrolysis of lipids. Likewise, you have amylase involved in hydrolysis of polysaccharides, pepsin, trypsin, chymotrypsin, carboxypeptidase, amylopeptidase. They are in protein hydrolysis. Likewise, you have nucleases, polymerases. You have enough example to say there are various classes of enzymes. We have hydrolases, lyases, ligases, polymerases, oxidases, oxidoreductases. Different classes of enzymes are there. You can imagine the number of enzymes involved in biological process. So proteins as catalysts, they are catalytic in function. They are involved in regulation, means they act as biochemical messengers. They pass on message. They are referred to as hormones. For example, insulin is involved in glucose metabolism. So insulin is a polypeptide. It is made up of 51 amino acids. Insulin has two polypeptide chain. One is with 21 amino acids, the other one is with 30 amino acids. The two polypeptide chains are linked by disulfide bonds. So that polypeptide is involved in regulation of glucose. So when there is excess glucose in blood, that should not be wasted. That should not be simply excreted, sent out. It has to be stored. So there comes the role of insulin. So release of insulin in an active form, when there it is in blood, means immediately another function starts. That is, all that excess glucose will be converted to glycogen. So it gives that message. So in the blood there is excess glucose, so that should be immediately converted to glycogen. So that message is passed on by insulin. In that way, there are several hormones involved, and under hormones we have different classes, and one class is protein hormones or polypeptide hormones, which are regulatory in function. Structure, they act as proteins acting as structural materials. For example, keratin in hair. Keratin is present in hair, horn, hoofs, feather, like that. It acts as a structural material. Even collagen also is an example. Actin, myosin, the muscle proteins involved in muscle contraction and relaxation. So they are also proteins. And here histone, it's the one which stabilizes the structure of DNA, stabilizes that double helical structure for protein for DNA. It's the histone as structural material. Transporting agents, they act as carriers. I already referred hemoglobin, involved in transportation of oxygen. It transports oxygen from lungs to all cells in the body, and simultaneously in the cell, in various biochemical process, carbon dioxide evolve that should not be allowed to stay there. It should be sent out. So that should be taken by the same hemoglobin. It binds to it, becomes carboxyhemoglobin, and it drops that carbon dioxide in the lungs, from where it gets expelled. And there is one more involved in as transporting agent or carriers, what we call it as cytochromes in the electron transport chain. They act as electron carriers, and many are involved as in transporting materials across membrane. So here in the diagram, it's represented how they act as channels, channeling the substances across the membrane, protein channels. And proteins are involved in defensive action. So we are living in a system with COVID-19 or corona pandemic. So today, how immunity is important, everybody have understood about immunity. It's the one, it's inborn, or it gets developed, the body, the system, it establishes that this particular property, what for? To fight against the foreign bodies, what are known as antigens. So the foreign substances, we are exposed to various types of attacks through various media. The foreign substances in the sense, those are not supposed to be in the body. So they gain entry into the body. Once they gain entry into the body, like coronavirus, when they gain entry into the body, they start utilizing our body machinery, and they multiply like anything and cause damage to the system, damage to the life. So therefore, they should be destroyed immediately. So proteins come into help here. Antibodies, they are referred to as antibodies, which are synthesized in our body against those antigens. So these antibodies are proteins which are defensive in action. So through nervous coordination, there will be recognition. The receptors will be there. They identify this. There is something which has entered the body which should not be there in the body. Immediately from WBC, these antibodies, the proteins are synthesized. They attack, they destroy the antigen, the foreign body. Excess of antibodies will be synthesized and they will be in the bloodstream. And when there is attack again, immediately they take care of it, take care of the attack. So in that way, this system, inborn system, is what is referred to as immunity, and it's the antibodies, which are proteins, which maintain this immunity. So actin and myosin and the structural materials itself I mentioned, they are contractile proteins in muscle contraction. I will not go to the details. There are many proteins which act as receptors for recognition, also act as secondary energy source. So this happens only when there is no carbohydrate intake. For example, insulin, I told you how it's involved in glucose metabolism. So when that activity is not maintained it properly, insulin is not synthesized in substantial amounts, there is some problem with respect to mechanism of release or its function, then there will be that excess glucose will not be converted to glycogen, it will be simply excreted, and this abnormality, what we call it as a disease in the name diabetes mellitus. This disease is referred to as diabetes mellitus. So during diabetes mellitus, when for energy, glucose is not available for energy source, then what happens? Proteins will be used up as source of energy. So in absence of carbohydrates. So in that way, they act as secondary energy source. Or during starvation also. During starvation, when you are not consuming any diet, any food, so then proteins serve as source of energy. The next one is lipid as biomolecules, mainly composed of carbon, hydrogen, oxygen, and no doubt their organic molecules insoluble in water, include fats, oils, waxes, phospholipids, steroids, glycolipids, and so on. The structure is, the basic component is a triglyceride. What is a triglyceride? It is a triester. What is an ester? An organic compound is classified as an ester when it's formed by the reaction between an alcohol and a carboxylic acid. So the product of the reaction of carboxylic acid and alcohol is ester. And here the triester is from glycerol, which is a trihydric alcohol made up of 3 hydroxyl group, and fatty acid, which is a monocarboxylic acid. So the reaction between these two results in a triester, what is known as triglyceride, because it is 3 fatty acids plus 1 glycerol, 1 trihydric alcohol. So that gives rise to a triglyceride. So that is the component of oils and fats, and many are phosphorylated, remain in phosphorylated form, what we call it as phospholipids. Here is a complex molecule cholesterol, the structure which is given here. And the hormones itself, I told you, we classify them as polypeptide hormones, peptide hormones. Sorry, under hormones, I didn't mention about 2 peptide hormones naturally occurring one, oxytocin and vasopressin. Both are nanopeptide, and oxytocin is involved in contraction of uterus during childbirth, and vasopressin in maintaining or reabsorption of water in kidneys. So like that, there is another class what are known as steroid hormones. So why we are referring to them here? Because basically they are lipids. So examples for steroid hormones, progesterone, which is responsible for changes in menstrual cycle and differentiation factor for mammary glands, aldosterone, which raises blood pressure and fluid volume, increases sodium uptake, testosterone, which is a male sex hormone, estradiol, an estrogen, principal female sex hormone, and cortisol, involved in stress adaptation, and so on. So in fats, in the fatty acid part, if there are multiple bonds, so they are referred to as unsaturated fatty acids. If there are only single bond, we refer them as saturated fatty acids. Importance with respect to lipids or their biological role, mainly they act as source of energy. Of course, carbohydrates also act as source of energy, but the energy supplied by an equal quantity of fat is twice than that of carbohydrates. So if we administer 1 gram of carbohydrate and 1 gram of fat, so 1 gram of fat will be giving double the energy supplied by 1 gram of carbohydrate. So they also act as source of energy in addition to carbohydrates, and they act as solvents for certain vitamins. And the vitamins at the end, I am going to mention about it. There you have the role of fats as acting as solvents for certain vitamins, what we call it as fat soluble vitamins, and they act as thermal insulators. Beneath the skin, we find a layer of fat. What for? To maintain the body temperature. So it acts as thermal insulator. And on vital organs like kidney, liver, heart, and so on, we find fat deposits. What for they occur on these organs, vital organs? They act as shock absorbers. So they protect these vital organs from their external shocks. And in the cellular membrane also you find phospholipid bilayer. That's very important with respect to membranes. So this I mentioned, shock absorber, good insulator. Next. And lastly, we have nucleic acid. With respect to importance, we say they are the one which are required to store the information and carry the information from generation to generation. And a study of proteins, I mentioned it, how proteins are synthesized. For example, when insulin has to be synthesized in the body, what all amino acids are there, what is the sequential arrangement, that information is available in DNA. And RNA, they carry that and help in the synthesis of protein like insulin. So after understanding the importance of proteins, you have to understand the importance of nucleic acids. For all such synthesis of protein, nucleic acids are very much essential. So there are two main: deoxyribonucleic acid, DNA, ribonucleic acid, RNA, of course, made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Next. So when a sugar, so there are three components in the monomer of DNA and RNA. Nucleotide, DNA and RNA is made up of nucleotide. Both DNA and RNA are polynucleotides. What is nucleotide? Nucleotide is phosphorylated nucleoside. What is nucleoside? Nucleoside is obtained when a base, purine or a pyrimidine base, coupled with or associated with a ribose or a deoxyribose sugar. So here in the diagram, you can see the three components of the nucleotide, A, G, T, C are the bases, right? G and C are the purine bases, A and T are pyrimidine bases. There is one more, uracil. It is present only in RNA, while thymine is present only in DNA. That's the difference with respect to RNA and DNA. So the nucleotide is formed by a combination of these three. So three components are here. The base gets associated with sugar. Sugar gets further gets phosphorylated. P is the phosphate group. So gets associated with the phosphate group. So that is what is called as nucleotide. So DNA and RNA are made up of such nucleotide. Yes. So the nitrogen base with sugar and with phosphate group. That's the structure of nucleotide. This again gives the same information. So this is the arrangement of nucleotides in DNA and RNA. So how and where they are. So in the nucleus, so you have chromosome, chromatid. So there you find so DNA with the structure shown here, a double helical structure, the two strands being held by hydrogen bonds. Can you see the hydrogen bonds between the base pairs, between the bases? Always the pairing occurs, the hydrogen bonding occurs between A and T, G and C. The three hydrogen bonds are between G and C, two hydrogen bonds between A and T, adenine and thymine, and guanine and cytosine. Purine, the base pairing is between purine and pyrimidines. Yes, see here. Adenine pairing with thymine, two hydrogen bonds. Guanine pairing with cytosine, three hydrogen bonds. This repeats in the chain, polynucleotide chain in DNA. Yes, it's the same information. And lastly, vitamins, they are also required for normal growth and functions of the body. But the amount of vitamins required is very, very less. They are required in tracer amounts. But if they are not present in substantial amounts, in tracer amounts, that leads to various problems. Even when they are present in higher amounts also, that leads to various problems. So if they are not present in substantial amounts, they lead to various disorders and various diseases if they are present in higher amounts, what we call it as hypervitaminosis. So we classify vitamins to main classes, water-soluble vitamins and fat-soluble vitamins. So important daily vitamins, along with that minerals, are also very important for normal health and growth. However, they are not considered as biomolecules. Vitamins, of course, they are biomolecules. And vitamin, there are several classes under, there are different types of vitamins under fat soluble and water soluble vitamins. So all are very important. And when they are not present in desired amounts, leads to various problems or disorders. So thereby, along with these dietary substances, carbohydrates, proteins, lipids and so on, we also should consume vitamins and as well minerals for the normal growth and functions. So I conclude with this. If you have any doubts, any questions, any clarifications, you can raise it now. Now the session is open for discussion. Thank you all. Thank you very much.